Today's webinar features Sarah Bentil as moderator and Hazel Screen of Queen Mary as speaker. Sarah is a faculty member in the Department of Mechanical Engineering at Iowa State University. And as you probably know, her work focuses on soft tissue and biomaterial mechanics. And Sarah will be moderating and introducing the speaker today and keeping track of time. Feel free to put your questions towards the end in either the chat or the Q&A box. And then Sarah will read them and discuss and share them with Hazel. That being said, thank you, Sarah, for agreeing to moderate, and Hazel for agreeing to speak today. We're very excited to hear about the latest developments in tendon and collagen and all sorts of related topics. So with that, thank you, and I'm looking forward to the webinar.
Thank you. Thank you, Professor Buehler, and thank you very much for your kind invitation to serve as the moderator for this JMBBM Frontiers webinar. I'd like to extend a very warm welcome to everyone this morning or afternoon or evening, depending on where you are, who is viewing this Frontiers webinar today. Right now, it is a great honor and my pleasure to introduce Professor Hazel Screen. She's a professor of biomedical engineering and head of the School of Engineering and Material Science at Queen Mary University of London. Professor Screen's research centers on healthy and pathological tissue structure, function, behavior, and its interplay with mechanobiology. She has a particular long-standing interest in tendon and ligament and leads a highly multidisciplinary group which spans human and animal in vivo and in vitro studies of tendon function and injury, taking a multiscale approach to exploring tendon mechanobiology. Professor Screen also leads the United Kingdom's Organ-on-a-Chip Technologies Network, and she also co-directs the Center for Predictive In Vitro Models at Queen Mary University of London, within which she leads a research group specifically focused on developing novel in vitro models of musculoskeletal tissues, which integrates her expertise in mechanobiology and structure function into new in vitro models to explore health and disease. Her presentation for today is titled Tendon Structure-Function Relationships, Health, Disease, and Aging in the Interfascicular Matrix. Professor Screen, we're looking very forward to your presentation. The virtual floor is now all yours.
Thank you very much, Sarah. That's very kind of you. And what do we say? Good morning, good afternoon, good evening, whatever time it may be where you are. And I think as we were just coming online, we were commenting on how wonderful it is to be able to do this type of event where we can really get together in a way that perhaps we hadn't traditionally done. To my mind, it's one of the very few benefits that's come out of the last really tumultuous year for us all. But congratulations on a fantastic JMBBM webinar series, and it's really lovely to be able to watch those, be it live or be it on YouTube later. Anyway, so thank you for the offer to be here. It's really exciting to chat a little bit about some of our work in Tendon and really I want to take you through a bit of a story as to how we started to explore and understand something about the interfascicular matrix in particular. And my background is as a mechanical engineer. So I guess if you said, where's my initial interest, it really sits around the field of tissue mechanics. So very much in the JMBBM area of exploring and understanding the structure and the organization of tissues, perhaps how they provide that tissue's mechanical function. But my interests really tend to interplay that with the mechanobiology side of things. So understanding something about how that tissue is structured and organized and how that implicates the cells within the tissue. So we know the cells respond to mechanical stimuli. That's the fundamental process that controls healthy turnover, disease, degeneration, anabolism, repair. But of course, those cells are responding very much to the mechanical stimuli that they perceive. And that is in turn affected very much by the structure and the mechanics of the tissue at a local level. So I'm really interested in the interplay between these two. And indeed, I think it's a good point to just highlight, for my mind, the real crucial importance of, if you're looking at this biomechanics area, taking multi-scale approaches to understanding this. So I've talked in the centre here, haven't I, about starting from a a tissue mechanics perspective and understanding within that tissue the nano, the micro, the macro response, but on a bigger scale, understanding how that implicates the cells, working down some of the signaling pathways and moving your way up through the hierarchical levels to understand how that impacts body mechanics. And I want to try to sort of take us through some of that area today. So let's introduce tendon then briefly. So I think we all know tendons are there. They transmit muscle force to the skeleton. And I've always sworn never to give a talk without this slide, but it's fundamental actually to everything that I'm talking about, really understanding this structure and how it provides function. This is a kind of schematic of the hierarchical structure of tendon. So here's your individual collagen molecule, nearly all collagen type 1. It builds to make fibrils. You see this kind of fibril in the basic collagenous unit in nearly all of our tissues. So this is looking at that hierarchical structure of tendon. And so here we are, we're building from that individual collagen molecule, you can see there, to make fibrils. So here you can see these are sort of classic fibrils as you'd see in most musculoskeletal tissues or any connective tissues really. In tendon, it keeps building. So we go through and they aggregate into fibers, as you can see here. So these are the cells in white separating fibers, aggregating again to make fascicles. And fascicles, you can see here, you can dissect. They're visible to the naked eye, so sort of 300 micrometer units, aggregating again to make the entire tissue. So this structure is really collagen rich, and sort of 80% of the structure, collagen type 1, aligned in this very ordered hierarchical manner. Each of these levels, the collagen units are held together by a proteoglycan-rich, non-collagenous matrix. So we're really looking at a very collagen-rich, multiscale fibre composite material. And one of the reasons I'm particularly interested in tendon is really trying to understand something about how we manage tendon injury. And that's tendinopathies, I think really we're starting to increasingly understand more about the socioeconomic impact of some of these diseases. So we know tendinopathy is chronically debilitating. It really has a very substantial impact on quality of life. And you see there's a very recent paper that's really been trying to look and explore that in a bit more detail. The prognosis is very poor. We see persistent pain. Interestingly, we see this isn't just a sporting issue. There's an increasing percentage of the sedentary population that are suffering from tendon injuries as well. And certainly a study a couple of years ago looking at prevalence indicates that it is beginning to outstrip osteoarthritis. So we're seeing a real issue here with a substantial socioeconomic impact. And if you take a look at the structure, here we go, is a sort of a really nice little schematic from this paper by me that looks at a healthy tendon and a tendinopathic tendon and tries to kind of give an overview of the differences between those structures. And look, this is this beautiful aligned collagen that you can see in a nice healthy piece of tissue, and the cells you can barely see aligned along those fibers. This is what you get in tendinopathy. This is a histology section. Here's the sort of schematic. We can see this highly disordered collagen. You see an increase in the cell population. The cells are rather bigger and more rounded. We tend to start to get capillary ingrowth into that structure. We see something that looks far, far less ordered. And if you were to go to your clinician with a tendinopathy issue, you might commonly see something like this, where you can really see that thickening of the tendon, degeneration within the Achilles. This is the back of the ankle and the Achilles tendon here. And if you were to do, say, ultrasound scan, this is Doppler ultrasound, you can see the colours showing vascularization, so blood flow through that tendon that you wouldn't normally expect to see. So I guess really the sorts of ways I want to take that tissue mechanics, mechanobiology area is to ask things like, how is it that multiscale mechanics might drive healthy or pathological tissue function? And to understand something about the interplay between the matrix mechanics and the mechanobiology that drives disease. And as we started to ask these questions about structure and function and injury, it's notable that we most commonly see injuries in energy storing tendons. And energy storing tendons are tendons like the Achilles. The name is a giveaway. They're ones that store energy during use to improve efficiency of movement effectively. And we can see this here in the Achilles tendon. As we run, it stretches, it stores energy, it recoils like a spring, and it improves locomotion efficiency. And we wanted to ask, why is it that we sort of see more injuries in these tendons? What's special about energy storing tendons? What allows them to perform that function effectively? So started to compare, for example, here, the Achilles tendon with, this is the tibialis anterior that runs down the front of the foot, which is what we would call a positional tendon. Tendons that aren't energy stores tend to just position limbs, they're positional tendons. And certainly we've done a lot of this work in human, and I'll come back and show you some of that data, but I want to take you through a story to begin with, looking at the equine model and some of the studies we did here to explore something about energy storing tendons and injury in this model. And this is a really nice model to explore. So the SDFT is a superficial digital flexor tendon, is like the Achilles tendon that runs down the back of the horse's leg. The CDET, the common digital extensor tendon, is the positional tendon that runs down the front. And these are beautiful, really old, but beautiful images from the Wellcome Trust Library, just showing a horse galloping, cantering, in fact, and just looking at that movement to give you an indication of why the SDFT is such an interesting energy storing tendon. And if we look at this leg here as it lands, here, this bit here is the fetlock joint, which you can see at this point here. As the horse's weight goes onto that leg, you see the fetlock joint here drop right the way down to the floor. And as that horse's weight comes forward, you can see that really huge stretch down the back of the leg, which is through that SDFT tendon. And as the leg lifts, that recoils and propels the horse forward. Indications are that it's something like a 27% increase in efficiency of locomotion for the horse. This is one of the reasons horses can run faster than us. So it's a really interesting model to explore, an extreme version of energy storing to understand the difference in structure and function. And indeed, if we just take an SDFT and a CDET tendon and pull them to failure, we can see immediately this difference in their behaviour. So stress-strain curve, we can see here the energy storing tendon is more extensible than the positional tendon, and it's less stiff. If we do cyclic creep testing, so we cyclically load to a set force until that sample fails, again, we can see here cycles to failure significantly higher in that energy storing tendon than in the positional tendon. So as a global picture, we see energy storing tendons are less stiff, they're more fatigue resistant. Okay. So the sorts of questions we might then ask is, well, why? What are the structural changes that might drive those different mechanics? And can we start to understand then if there's something about poor structural optimization that leads to injury? And that's what got us started in this area. And one of the first things we did is say, well, can we investigate something about micromechanics within fascicles? So taking fascicles and exploring the mechanics at these sort of micro levels here. And we set up techniques to do this many years ago now, where really you can take viable tissue samples. We can put them into a custom-made rig here. So I built this, I don't know, about 2000. So we can put this onto a confocal microscope and take living tissue samples, stain something within that sample. Look, this is the cells, so you can use them as sort of little particular markers of how the strain is within the sample. Or we've done the same here. You can put grids on and you can look at how that grid deforms, but you can strain that sample and look in real time at how it is deforming. And if we do it, this is an example of what happens if you compare a fascicle from an energy storing and a positional tendon. And you can see just by inspection that we see this rather different shape to the grid after deformation. And we said, well, how can we sort of put some numbers on and start to quantify what we can see? And you can measure the local strain, so how much strain we get along this grid, the transverse strain, which is interesting to get a feel for the Poisson's ratio within these samples, but also this measure of fibre sliding. So over here that you can see this line is kind of broken. We can see how that fibre is sliding, or we can look at rotation so that you can see here it's no longer aligned, it's twisted, and we can measure that. We actually see local strains are really quite small. We see really large transverse strains. It's quite interesting, Poisson's effects. But I'm going to concentrate on these two, and I've given you a little hint of what we might see. I'm sure you can see for yourself in the pictures. If we bring all of the data together for these samples, I'll commonly draw graphs like this, where we look at the applied strain against the parameter of interest. This is the amount of fibre sliding. We see as we increase the applied strain, You see, very little happening here with the energy store. Positional tendon is showing increasing amounts of fibre sliding. By contrast, with increasing applied strain, it's rotation we see, reorientation in the energy storing tendon. So these fascicles seem to be showing different mechanisms by which they stretch when we load them. And that's interesting, but I always worry a little bit about looking at stretch in isolation, because you don't know if you're looking at damage mechanisms, you don't know if you're looking at something physiological. So the next question is always to say, well, what happens when you take the load off? Do they recover? Can we understand something about that response? And certainly we can see that this isn't just pure damage. They recover reasonably well, better if you leave them at a period of time to recover, so you can see that viscoelastic effect. But we do see that even fibre sliding or rotation, the energy storing SDFT recovers better. And that's perhaps not surprising. This is load-unload curves for 10 cycles where you can see how tight they are in the energy storing fascicle compared with the positional, where you can see increased hysteresis. So we seem to see better recovery from loading in energy storing tendon fascicles as well. So it builds a picture of fundamentally different mechanisms by which these similar looking fascicles actually function. In the energy storing tendon, we see rotation seem to dominate as the way to allow that extension and bounce back, whereas in the positional tendon, the extension seems to occur through sliding, but there's poorer recovery from that. It's a more viscoelastic response. And it leads us to hypothesize that SDFT fascicles have a helical component. And certainly if we look, here's an EM image, you can see that helical structure. And we've been starting to look and explore a little bit about how we might model this and understand a bit more about its impact on the tissue. But I want to move on to sort of the next level of the hierarchy and really what we looked at next. So we'd seen this difference in the fascicles, which was interesting. But if you actually take those fascicles and just pull them to failure, down here, we see exactly the same mechanical behaviour, which is not desperately exciting. But it is kind of interesting if you think the fascicles from the energy store and positional tendon seem to behave exactly the same. The whole tendon, if you remember back, we saw this clear difference in mechanical behaviour. So we've got us starting to ask a little bit about how, why, where is this difference in mechanics coming from? Because it doesn't seem to be from the fascicles themselves. And if we take a look at a schematic here, of course, here are the fascicles embedded within the entire tendon structure. If it's not coming from these fascicles, it's going to be coming from the matrix around them. Here, the interfascicular matrix. And it got us started exploring this structure. And you can see it, just to give you a little sort of feel for what it's like. This is just H&E staining, so this is the collagen within fascicles. You can see there's fairly rather different disorders. This is proteoglycan rich, you get purple colouring for proteoglycans. We can see them in the interfascicular matrix. Here, this is DAPI for the cell nuclei, so we can see these nice elongated cells within fascicles, very different rounded interfascicular cells. So my interest being mechanics, one of the first things I wanted to do was really say, can we explore the mechanical behaviour of the interfascicular matrix and develop some sort of mechanisms to test this? And this is what we did. So well, if you can take out two fascicles together as a unit, so here they are schematically in yellow, you'll have those connected by that line of interfascicular matrix. We could cut off opposing ends, so we can keep a defined length in the middle. There's our sample. We've effectively got something that we can shear test. So we can pull the two ends and explore the response of the interfascicular matrix specifically. And here it is, so you can sort of pictorially see what that looks like. We can grip it, we can cut those, we can pull that apart. We can explore the mechanics of this region here, compare it with the mechanics of the individual fascicles themselves. And if you do those sorts of tests, this is what you see. These are force extension curves for the energy store and the positional tendon. And I kind of had a wave of emotions with this one. I think my first thought was, wow, that's remarkably strong. If you take an individual fascicle and you put it to failure, I'd say roughly around the 3 newtons is the sort of failure force that you'd be looking at. So seeing this over a newton that's a sort of failure force surprised me. It indicates there really is some substance to the mechanical properties of this gel-like material that holds the fascicles together. The next thing I thought was a pretty sad conclusion, that if you look at this endpoint here, really they fail at exactly the same force, exactly the same extension. I couldn't see anything particularly interesting there that might explain some of these differences at the tendon level. But then, of course, when I actually started to think about it, you realise that this is complete tendon failure up here and everything falls apart. If we're looking at the kind of functional range of this tissue, we're looking somewhere around here where we start to see this difference in the mechanical behaviour of the two tissues. And indeed, this is an example curve, but we can quantify that if we look across all the samples. If we were to look at, 30%, maybe roughly here, 30% of the failure force, we can move along and say how much extension is there at the interface. You can see in this example, there's more in the energy store than there is in the positional. Here it is graphically showing all of that data together. There's significantly more extension at the interface, the large majority of the way through that curve in energy storing tendons than we see in positional. We see this extended toe region in the SDFT IFM, which seems to provide less resistance to fascicle sliding during this physiological loading region, perhaps enabling that tissue to stretch more easily. As I said before, that's interesting, but is it recoverable? Is it meaningful? Well, we did some cyclic creep tests to try and explore this material in a bit more detail. And this is a pretty aggressive test. So this is loading to 50% of the failure load and cyclically taking it to that value and looking at how it responds. This is the curves, loading, unloading curves for the first 10 or so cycles. You can immediately see they're tighter here in the SDFT than the CDET. So we're seeing more elastic behaviour. We're seeing significantly more cycles to failure in the energy storing tendon than the positional, so more fatigue resistant behaviour. So we certainly are seeing mechanical specializations in the IFM. So the other side of that coin is to say, well, how about something about the composition and the structure? Can we bring this together to understand a little bit more about what's happening? So we started to look at compositional specializations. So one of the first things we did was just stain proteoglycans and see what we could see. And this is kind of interesting, really. There's definitely more proteoglycans, so it shows up, remember, in this purpley colour in the H&E. There's certainly more of it in energy storing tendons, and certainly more of it localised to the interfascicular matrix that we can see here than you see in the fascicles themselves. This is perhaps a little bit surprising. You wouldn't potentially expect this when we're talking about an elastic recoil behaviour. So we wanted to probe a little bit more and understand sort of specifically what proteoglycans were driving this. And these are probably the three of the classic ones you might look at when you start to talk about tendon: decorin, fibromodulin, lumicin. Certainly with all of these, we just see a homogeneous distribution across the fascicles, across the IFM, and really no differences if we looked at the energy store, the positional tendon. So it didn't seem to be these particular proteoglycans that were influencing the response. Where things got interesting is when we looked at Lubricin. So here again, we can see it in this brown colour. Lubricin, the name is a pretty good giveaway. It's a lubricator. And here it is localised to the interfascicular matrix, where we see notably more, but significantly more in the energy storing SDFT. So perhaps it's there to lubricate or enable that sliding behaviour. Of course, we've been talking about recoil. So the next one we really wanted to explore was something about elastin. If we just look at overall content with a standard sort of assay, the fasting assay, we can certainly see there's significantly more elastin in the energy store than the positional tendon. Still isn't a lot, 1, 2, 3% sort of elastin content in this tissue. But look where it is. So here's that interfascicular matrix region, the elastin in red, entirely localised to that interfascicular matrix region as well, perhaps there to enable that recoil behaviour. So we're starting to piece together this sort of composition to provide this specialist mechanics and enable function. Certainly we've done a fair bit more exploring the proteome of this structure in detail. I don't want to talk about it a huge amount, except to say this repository is available now if it's something that is of interest. We used laser capture microdissection, so it's a lovely way that you can take sections, frozen sections through a tissue, use a laser to cut out bits of, this is just the IFM you can see here, or just cut out a bit of the fascicle, put that through mass spec and look at the proteome. Certainly the IFM was a more complex proteome, sort of get a feel for what's there. It won't surprise you, there was vastly more collagen 1 within the fascicles, But we saw a lot of minor collagens in the IFM, lots of the collagen 3, that seems to be where it's housed. I said there's a repository of that data to explore if that's of interest. But just for today, I wanted to focus on one thing that was really interesting to me when we started to look at this data. So one of the things you can, when you do mass spectroscopy, you effectively take your sample, you digest it in trypsin, so you get this series of peptides, as you can see here, and then you can look at the peptide sequences and identify the protein that they are, and that's how you effectively look at what's in the structure. But if you look at where that peptide is cleaved, you can use that to explore something about matrix turnover. And that works because if you have cleaved a protein with trypsin, it's very defined where that trypsin cleaves. So the point at which that peptide sequence is broken, we know where that will be. So you can identify if something is a tryptic sequence. If it's a tryptic sequence, we're going to ignore it at the moment because it's told us that within our mass spec preparation, the trypsin's broken it down and we can just explore it with proteomics. But we found a series of non-tryptic lengths of peptide as well. And if that peptide is non-tryptic, it tells you that it's been broken down, it's been cleaved before we started this trypsin process, which effectively is telling us that it's been cleaved in the body, which is an indication of natural degradation or natural turnover. So the more non-tryptic or semi-tryptic run-ins and non-tryptic peptide that we see, the greater the amount of natural degradation we're seeing within this sample. And we were seeing a lot more in the way of non-tryptic or semi-tryptic peptides in the IFM, indicating a faster turnover rate. And I said I'm really interested in this interplay between the structure and the cells, and this is a really nice sojourn to ask a bit more about those cells. Here they are. Here's the sort of elongated cells within the fascicles. Here's this larger number of rounded cells in the IFM. Certainly we can see there's more of them. The proteomics suggests they're more active. We had a look in a number of other ways as well, and one of them was to look at something called C12C, and that is a marker of collagen being broken down. So you can see where there's a breakdown, which of course is indicative of a turnover process for that particular collagen. And this is just a bright field section through the tissue, so these are the fascicles, so you can just see where that IFM region is. And if I turn to the immunofluorescence to show you where the C12C is, there it is, entirely localized to that interfascicular matrix region. So we seem to see a greater activity in this region also. So perhaps you can just sort of bring that together and just reflect there on really a series of specializations in energy storing tendons centering around the interfascicular matrix. So we saw this specialized mechanics being just in the IFM region, a composition to enable that, and a cell population that seems to be more active and encourage greater turnover as well, perhaps to maintain that structure. And of course, seeing that difference in cell population leads to say, well, what's happening in injury? Can we understand something about the injury response in these samples? And to start to explore this, we said, well, let's just in the first instance look at explants of tissue. So we can take living, viable tendon explants. We built this a number of years ago as a sort of a large loading rig, so we can mechanically stimulate a number of these at the same time and look at their response. And here it's the same sort of bright field I showed you earlier. So this is the fascicle structure. Look, here's this interfascicular matrix in an unloaded sample. This is what it looked like after sort of 1,800 odd cycles of loading. I think it's important to contextualize this because you want to get a feel for how much damage you've created and where you've created it. Certainly, look, we can see, I don't really see a huge amount of damage within that collagenous fascicle structure. That disruption was nearly all localized to the interfascicular matrix region. And when we then started to look at a number of different markers from the cells in that region, this is the first one we looked at, COX-2, a marker of inflammation. We saw that that response was entirely focused within the interfascicular matrix cells. Here's a few more pictures sort of from the bigger studies. So actually we can look at 300 cycles, 1,800 cycles, so we can increase the degree of loading. Again, staying at a point where we're not pulling this to complete fracture. It's very early damage. We see that damage accumulating, increasingly disruption just in the IFM. And we start to see an upregulation as well here of MMP3 and MMP13. So these are markers of ECM degradation, collagen degradation. So we're seeing this cellular response specifically to the IFM region. So it seems to suggest that not only is this IFM key for the function of tissue, but it seems to be the real region where we see the early response to injury as well. So one of the next things that we did, trying to explore this in a little bit more detail, said, well, can we understand something about age? We know injury risk increases with age. If we're earmarking this as the possible region that's the driving injury, can we see changes with aging? So we went through really, and I'll take you through the same test a bit quicker to see what happened. We carried out the same sort of IFM test to see how that responded. In the positional tendon, really nothing changed with age in the IFM response. If we look in the energy store, we saw that elongated toe region, here it was in the young horse, was lost with age. I think it's a terribly depressing graph, this one. Look, as we go up with age, we see this steady decline of that ability to have that toe region, that extension at the interface. And obviously 20 is elderly in horse years, so we shouldn't feel too bad about this. But we can see that steady decline in IFM mechanics. If we look at that fatigue properties as well, that recoverability, this was the graph that I was showing you earlier. This is just max and min this time on the cyclic creep curves. But this was the one where we were seeing greater cycles to failure with the IFM in the energy store. We see that number of cycles drops right down here, sort of 200 odd, matching the CDET with age. So that increased fatigue resistance in the young SDFT IFM is also lost with aging. If we look at the composition, it's just overall elastin content. Here it was, remember it was higher in the energy store than the positional when we were young. We see that drop down with age in the energy store in tendon. And if we actually look at that, remember it was stained in red, we start to see real fragmentation with aging within the IFM, of course. It's where it's all localized. If we go to the proteomics and look at some of the changes with aging, there are a few, again, that's sort of there if you want to explore it. Fibromodulin certainly changed quite notably with aging. But the point that I wanted to draw on today was that that non-tryptic peptide number, that measure of turnover, we saw that decreasing significantly with aging in the IFM only. So we're really, again, pointing to that sort of loss of turnover in that region. Okay. So we can sum those aging changes together again now too. Mechanical structural changes only seem to be evident in the energy storing tendon, and were nearly all localized to that interfascicular matrix region. We saw the lubricin elastins reduced, the extensibility was lost, and that fatigue resistance was lost. We start to see fewer cells, less matrix turnover, seem to see a reduced capacity to repair damage. So we've kind of pulled together a story in this equine model of what might be happening, and I hope I've pointed you towards the IFM and why we find it so interesting. I promised at the start that I would talk very briefly about the human model as well, and certainly we've recently here at just in Biorexiv, so if you wanted to look at the full set of human data that's now available to see, we said, can we do these really similar tests, explore the composition, the mechanical behaviour of fascicles, IFM, whole tendons, comparing the human Achilles and the anterior tibialis, and see if we see this same specialism and the same changes with aging. I think broadly, as we can see here, this data is very consistent with the equine data. But here's the Lubricin localised, the Elastin localised, we see that same kind of behaviour. We see the same real lack of differences in fascicles. We see the same fatigue resistance in the IFM. It was notable that none of the differences were as extreme as we saw in the human model. Possibly quite likely because obviously it's really hard to be managing samples in this space. We've had to compare middle-aged and old, and certainly we don't have that young group. So we could hypothesize that we've already lost some of that specialism already. And of course, as I said at the starting point, I would expect that horse to be highly specialised for energy store, and why it was such a useful model to try to understand some of this, because I'd expect it to be more extreme. But certainly we're seeing the same sort of response. So moving on from here, it's really leading us to go down two avenues, two different hypotheses focused around this interfascicular matrix story and where the work is taking us now. We would hypothesize that tendon injury really originates from microdamage and a cell response in the IFM. And when we start to see downline collagen disruption and turnover and that tangled collagen mess, that isn't an initial response, it isn't a direct overload response, but that's a cellular response down the line after you've had this IFM cellular behavior in the first instance. Secondly, we'd hypothesize that those that are injury prone are going to have a less specialized IFM, so a structure that's less able to manage that effectively. And we've been using a range, again, back to home roots, multiscale sort of approaches to try and explore this in a bit more detail. And we've been asking a bit about, well, what in vivo models could we build, different approaches that would enable us to validate this or understand it in a bit more detail. So going up the hierarchy, but also going down and saying, how can we start to directly explore some of this mechanobiology or IFM cell behavior? So if we head upwards first towards in vivo models and show you where that's starting to head. And we've been looking at the rat's Achilles, really, as a model that has a really sort of a similar IFM, a similar tendon structure to our Achilles to allow us to produce an in vivo model to explore. Certainly the Achilles tendon, as you'll see in a couple of slides, we know it's attached to the triceps sura, it's attached to a number of different muscles. So they pull different parts of that tendon interdependently, so you can sort of have an interface between the parts of the tendon pulled by different muscles. We see that in the same way in the rat model. And if we explore the structure, we can see a lot of similarity here between the horse and the human that I've been showing you. So here's the elastin in brown, localized to the IFM. Here's the lubricin, localized to the IFM. So these same sorts of structural specialisms. This is a mechanical test of the IFM, or that interface between the sort of sub-tendons in the Achilles. We can see this same cyclic behavior, really similar response to the tissue. So we see that this is a consistent mechanical compositional setup to the equine and human data. So we've been trying to take this forward really as an overload model now to try to understand if this hypothesis that we've started to test in an explant, if we really see that when we test in vivo, when we've got the whole muscle tendon complex functioning in a more physiologically relevant way. So this is the rat Achilles tendon overload model that we've set up. So drawn in a little loading arm here, so you can see this is the sort of a small bows, so we can load. We can secure the rat leg here in the setup, so we can cyclically load. And we've just done a preliminary study to just give you, which I can show you now to give you a feel for where this data is going. So we've done one single bout of 100,000 loading cycles, and then either looked at the response immediately or culled the rat 48 hours later or 4 weeks later, and just used immunohistochemistry to see something about the cellular response, the tissue response. And certainly the first thing we can do is just look at the bright field. So this is very much as you've been looking at before, fascicles. Here's the, wrong one, point, here we go, fascicles, the interfascicular matrix, so you can see those different regions. It's useful to look at this. Remember, we can see if there's structural disruption. This 100,000 loading cycles in vivo, it's clearly not an extreme loading protocol. We couldn't do that and then return the rat to cage exercise. We can see it's not creating clear structural disruption in the samples, but we are loading and we can now start to look for a cell response. And these are the sort of initial images and what we can see. So in the first instance, we looked, as we had before, at MMP13, MMP3, so those two markers of matrix degradation. We've also got alpha smooth muscle actin, so that's a marker from the cells to say that they perceive load and they're responding to that applied load. And we've got IL-6 as initial marker of an inflammatory response within the tissue. So in the sections, these little white lines here are there to highlight the interfascicular matrix region for you, so you know where that is. We've got Hirsched here, so we can see the cell nuclei, so we can orientate ourselves a bit better in those samples as well. These first ones are just looking at the unloaded controls, and of course we did them at all time points, but effectively we saw minimal real cell response throughout perhaps a little bit of MMP3 with a consistent indication of some activity here. This is what happened when we looked at the loaded samples. So in the first instance, we've just got the MMP3, the IL-6 here. We're looking at increasing time. So these are the ones culled immediately, 48 hours, then 4 weeks. And we can see here a very clear upregulation of both MMP3, IL-6. That seems to become more diffuse as it's allowed over time. Even more diffuse by 4 weeks, it seems to be spreading through the matrix. If we look at the MMP13 then and the alpha smooth muscle actin, again, we can see this immediate response. If we look at the alpha smooth muscle actin, it is very much immediate and that dies away again. That's perhaps what you'd expect. This is the cells responding to that load immediately. That's a good indication that they perceived it. That goes away, we're not reloading. So that's a sensible response. But here with the MMP13, we can see that ongoing upregulation in these samples. So it's a really early indication, and we're in the middle right now of a sort of a bigger study to make a little bit more sense of this, link some of it to the mechanics and try to build a greater picture. But we might just start to map out these kinds of pictures that show over time this immediate cell response and then this upregulation and slow tail off of MMP3 and IL-6 and a slightly longer tail with the MMP13, certainly showing all of this overload response for the IFM focused. So linked to that, I said we also would very much like to get at this in humans somehow and say, how can we explore the response here? And I said I'd show you these kind of pictures of the Achilles tendon. Here they are, this idea that you've got multiple muscle bellies coming into the tendon. So you've effectively got sort of a series of subtendons, the bit of the muscle that feeds into that section of the tissue. And we can sort of take this tendon, and the hypothesis is, you know, if your IFM is not allowing, it's not allowing sliding, you'll be able to see that the whole Achilles tendon is straining homogeneously. You're not seeing subtendon sliding. It's been interesting, actually. There's been a bit of a growing interest in this for a number of years. This is some work from Laura Slane's PhD, which she did with Darryl Fellon a few years ago, really looking at non-uniform loading across the Achilles tendon just during different exercises and walking. And here she used an ultrasound probe to explore what was happening at the tissue, did frame-to-frame speckle tracking to explore the strains or the displacements through that tendon during loading, and came up with some really nice pictures here. So this is at the start of loading, this is at the end. You can see here the deep portion of the tendon stretches more than the anterior portion of the tendon. Non-uniform loading through the thickness of the tissue. What I thought was really interesting is that the non-uniformity was greater, a 16% difference between the two sides of the tendon in young individuals compared to middle-aged individuals. And there's a real growing number of studies that are exploring this, and it's really been very interesting to see this picture coming out. But where we really want to go with this is to say, well, can we actually find the slip planes? If we're really interested in this IFM mechanics, is there a way that instead of sort of globally looking at deformation, we can specifically identify slip planes themselves? So we've teamed up with Jeff Bamber, who works at the ICR, where he developed a technique called ultrasound slip elastography. And he really developed this for looking at slip planes in tumours. So here we are. This is identifying a tumour boundary where there's a clear slip plane, which is really important in managing tumours to know how easy they are to cut out or remove, to see how well adhered they are to tissue. And we've been working over the last couple of years to say, how can we translate this technique to be able to get this clear information about slip boundaries within tendon, be that the human Achilles, so here we are set up to look at that, or the horse SDFT as well. And certainly here's some of the sort of first data. You can see that Achilles tendon here, straining. You can see that in the B mode video. This is if you start to measure shear strains using that slip elastography method. And we can see it's sort of mainly blue as it's sliding under the skin, but this mixed colour is indicating non-uniform strains. Potentially we're starting to pull out some of those slip planes, and we're working to optimise that. But I've got just a couple of minutes left, and I do want to use them just to quickly go the other way, as I said, and try to link that in a little bit to the mechanobiology. And I think Sarah kind of introduced at the start that we're now working with the Centre for Predictive In Vitro Models. How can we use the in vitro side to explore this in greater detail as well? Well, actually, this is really challenging in Tendon because we really want to understand something about the IFM cells, see if we can manipulate their behaviour, but we really don't have any markers or any mechanism to extract these cells to explore them in detail. So we've really been starting saying, how can we get a handle, explore, understand something about the markers that define IFM population or a fascicle population and allow us to understand something more about their processes. And to do this, we've had to move back to the rat model and specifically the rat tail. And the reason for that is this is a sort of schematic of the rat tail. The tail has this kind of layer of IFM around a fascicle, which is very useful because we can see spatially that we can develop a sequential digestion protocol where we can digest off this outer layer, just get IFM cells, to discard the middle, just get xenocytes, and validate an approach that allows us to separate those two populations, to start to phenotype and explore them in greater detail. And here, if you just put them on plastic, they sure look different. To get us started, we're building a bigger picture of these and some of the differences between them. It's really very interesting to explore. Certainly our IFM cells really hate tissue culture plastic, probably because it's so stiff and they're used to this really soft gel-like material. So we're starting to build this picture. But where I really want to take this is to start to say, well, can we build tendon on a chip models? And for those that haven't really been introduced to the organ on a chip concept, the idea really is how do you build an in vitro model of that tissue? So you put it on a chip, but it's just in an environment in vitro that really allows you to explore something about the cell behavior, the mechanics, the phenotyping of those cells, how they might drive disease, how they might respond. And here we are starting to build a tendon on a chip model. And the idea is to house our IFM cells in a soft IFM-like matrix, put them in this bottom channel here, get our tenocytes on a nice aligned, stiff matrix in the top channel here. These channels are separated by a porous membrane, so we can keep the cells housed separately, and we can look at signalling between those two populations. We can apply strain, so there's a mechanism here to stretch this, so we can load those cells and look at their mechanobiology response. And so we just started to get this going, and I just wanted to put these in because I'm really excited to start to pick up data. Certainly these are just the tenocytes, starting to get aligned surfaces here. We can see them beautifully lining up. We can get these in the top channel of the chip, and here's our first data with the IFM cells here in red on the bottom of the chip, and you can see this very different sort of cobble shape that they're showing relative to these aligned FM cells in the other channel. And we're really looking forward to starting to use this to explore something about the cell signaling and see if we can really understand what it is that's driving that pathology. When those IFM cells first kick off the response, what is it that takes us to that point where we see this really disrupted collagenous structure? So I think that's hopefully a nice place to stop and just sort of given a bit of a whirlwind really around some of the work we've been doing, how really starting from tissue mechanics, starting from that structure, function, understanding, it leads into this hugely sort of multidisciplinary area of understanding a little bit more about how the cells impact and how we could eventually start to explore injury. And for us, why we're really very interested now in the IFM, and it's a key target for us to try and understand potentially how tendon functions in health, where and how disease develops, and if this is the kind of early responder that we might be able to target in some of our therapies. Okay, so on that note, I can't stop without saying thank you to a lot of people, certainly to our funding sources, but specifically for our researchers. Siobhan really took a huge amount of the load with the equine work, and she was worked with me for sort of 6-7 years as a postdoc and really took this forward, and Marta worked with us as a PhD student, leading that Elastin work in the equine model. Dharmesh and Eva really took that human data forward, and we were able to explore that in the human model as well, whilst Gamal is working now developing that sleepy elastography technique and starting to understand a little bit more about in vivo tendon mechanics. Connor has been driving the rat overload model with Antonis. I've got a picture of, he's down here. And Simon has come and joined us recently and is now leading the group, starting to build that tendon on a chip model as well. So this is probably our lead team, but certainly all of these individuals have been involved in different elements of the work. And here's the primary collaborators and groups. And I think I hope it has given a feel for just how multidisciplinary this is. And it needs to be to take this sort of approach forward. So Peter works as an equine vet, a matrix biologist. Dylan as a physiotherapist. Helen's a matrix biologist. Graham works very much in cell biology. Stephanie and Martin I've been working with in the vitro models, understanding some of that cell biology as well. Jeff Bamber I mentioned, who comes in and brings that ultrasound elastography element in as well. And Siobhan is now, I'm delighted to see in a fellowship post and taking some of the cell work forward a little further, whereas we've been working with Alicia to start to build in vitro models. Steve's been involved in looking at the Elastin work, and Hoop has some real expertise in the in vivo animal models and been working with us in the rat model. So on that note, I will stop and hand back to Sarah. Thank you.
Great. Thank you very much, Professor Screen, for this wonderful and informative presentation on structure-function relationships of fascicles and interfascicular matrix and energy storing tendons and the role in injury and aging. I learned quite a bit and enjoyed your talk very much so. There are several questions that have been generated in the chat and the Q&A, which I'll read to you now and kindly request your response. So the first question is from Babak Safa. And Babak says, thank you for the excellent talk. And Babak was curious to know a bit more about the equine model. In general, Babak suspects that it is less available animal model compared to the common rat and mice animal model. So what are the sources for the horses? And is it possible to do any in vivo or longitudinal studies on those horses?
Brilliant. Well, thank you. Yes, models. It's an endless headache. So the horse model, the good things with the horse model is that for the sort of waste tissue work, a lot of that early in vitro activity, it's easy. You can go to an equine abattoir, so we can just collect waste tissue from the meat industry. So we used to do that with sort of bovine tendons. We can do that with equine samples as well, and it's a good available source with sort of no issues there. For the in vivo studies, of course, we're linked in. So Pete Clegg works at an equine veterinary hospital. They see a lot of equine veterinary subjects, SDFT injuries. It gives us a really good in vivo source to be able to explore using ultrasound. But of course, you're looking at that model then very much like you look at a human. You can sort of use ultrasound, non-invasive methods really to explore the response. And to be perfectly honest, horses are far more annoying than humans when it gets to this point, because if you're working with a human, you can tell them to stand on one leg or jump up and down or do whatever it is you want, and it's much more controlled. That actually obviously becomes challenging with a horse. So it's a little bit hard, but you certainly can carry out these sorts of studies. And we've just put in a bid for some funding, fingers crossed, to try to carry out exactly that, some longitudinal studies where we say, could we look out at some injured horses, follow them over a year, try to look at how that IFM mechanic changes, if it resolves, if that links with pain or mobility in the horse, and try to understand some of that behaviour in greater detail. So yes, those models in those spaces are very, very useful. Of course, for an in vivo model, an overload model, it doesn't hit the bill, and that's where we need to look for something that enables us to explore those sorts of questions.
Great, thank you very much. We have another question from Hannah Isaacson. Hannah first says, fantastic talk, thanks Hazel. And then Hannah goes on to say, the in vivo rat overload model looks great. Could you tell what strain levels you aim for? And also, if you have explored longer term response?
Okay. And I'm going to get it wrong if I try to give you numbers, so I'm not going to. We're trying to bring the data together at the moment with the strain. Getting control, precise control of that model is a problem. It's not bad. We can define angle, and that's how we've done it, that we've defined the maximum angle and the minimum angle of load. And we've got a series, what we've done is stripped in cadaver models. You can strip it down and you can put a series of strain markers along the sample, along the tendon, and you can define the strain. I forget what those values are, so I'm not going to pretend I remember. It would be better if I look them up and I tell you later, Hannah. But we've sort of done some of that as an early validation of response, and we're trying to get in a range that we keep a reasonably consistent strain. It will not be exactly the same in every sample. That's not possible, but it's pretty good. So that was a bit about the strains. Have we looked longitudinally? Not yet. So that really is a preliminary study. The one we are in the midst of right now still only takes us out to 4 weeks, but we're trying to do that as a broader with a good number of samples, trying to look at a little bit more than just the immunohistochemistry. We'll look at some of the mechanics as well and see if we can get a slightly more robust response. We also need to do a series of sort of PCR measures, I think, really, so we can look at some of the gene expression, some of the protein expression, see if we can get a better profile. And my hope is once we've got that, we can pick where to go, because there's a few things we could do. We could go out longer, we could do a second bout of loading, and I don't know yet, and I'd rather sort of get this full data set and make sense of which of those is the most sensible thing to do to try to ask a bit more detail. But it's exciting to see it coming together. We certainly have got an age study, which we'll be doing in the next sort of couple of months. So we'll be able to compare the young and the old group and see how that relates to the equine data.
Very interesting work coming forward. So we have another question, this time from David Taylor. David states, great talk, Hazel, good engineering and biology. And the question from David is, will the structure of the SDFT change when its level of use changes? For instance, when the horse retires from racing?
Thanks, David. I have no idea. Probably. But one of the things that we don't understand, and I'm hoping we can slowly start to pick up, and it's one of the sort of things that we'll hopefully be able to link when we get the sort of the in vivo through to the in vitro, is to try to make a little bit more sense of some of these aging changes. Certainly we can sort of see these early bits of mechanical changes. We can see that just with the aging profile, we see cross-linking, and we're trying to piece it together. So can we look at the level of cross-linking and link that with the IFM mechanics and try to see what drives what difference in response? But we haven't really yet got a group where you've taken a horse that's being heavily loaded and then it suddenly stops and it changes. But you'd expect to see some kind of change. And certainly if you put that with the context that, we know that tendons change slowly, but if you move into elite sport over time, you seem to see these slow changes in the diameter of the tendon. It will slowly get a little bit thicker. People are reporting changes in mechanics, improved energy efficiency. It's all starting to indicate that you get slow change. But I think one of the keys is slow. And certainly I can't remember where, I think it might have been Jill Cook who I first heard say this, who said tendons don't like change. And it's true. If you change the mechanical environment on them, whether you overload or you underload, they invariably seem to get grumpy, and there's this time and this ability to respond that is not as easy as it is in the good old world of bone, where I think things are much more responsive and change better. But yeah, we'll see.
Great, thank you very much. And to be conscious of everybody's schedule, we have time for one more question, and this question is from Leela Nuri. And Leela's question to you, Professor Screen, is as follows. So in the clinical settings or in vivo condition, tendon is exposed to the combination of loads, for instance, tensile loading, shear strain, rotational load at the same time. So the question is, as stated, how does your work, where you're looking at single loading of the tendon, how does this apply to the real world?
Yeah, I think that's absolutely the key of why we had to move from an in vitro model, where you're taking a single, explant and cyclically loading it, moving to in vivo, because it's not even just that off-axis loading and everything. It's the muscle pulling separate parts of the tendon independently. All of that factors in, and that's why we're looking to try to provide a loading environment that's as close to physiological as possible, to try to make sense of how the tendon responds in that environment and whether the changes we see actually map across. But I think that's the science that you have to make sense of, isn't it, to a degree? You have to kind of find a model system, simplify to start to understand, be very conscious of the simplifications that you've made, but use that to try to unpick and make sense, and then work out how you can extrapolate back and provide confidence that what you're seeing isn't artifactual, but is actually a true response. So I'm hoping that's what we're really seeing, and that's why we've moved to that in vivo model, and it's encouraging, seeing really very much the same sort of response.
Great. Thank you very much for answering those questions, Professor Screen. And thank you again for such a wonderful presentation. I really enjoyed your talk. And Professor Buehler, the virtual floor is now yours. Thank you.
Great. Thank you so much, Sarah, and thanks so much, Hazel, for a wonderful talk and a wonderful webinar. We will be taking a break over the summer. We will be back in the fall with new webinars. And until then, I wish you a great summer and I hope to see you soon. Thank you so much.